My name is Marios Nikolaidis. I'm affiliated with Mayo Clinic in Rochester, Minnesota, and I work in the Department of Gastroenterology and Hepatology under the supervision of Dr. Konstantinos Lazaridis, who is a hepatologist and gastroenterologist. And our research is about studying the microbiome of PSC and PBC patients, and we're trying to identify through the microbiome certain aspects of the microbiome that may be used to help manage these diseases. So in order to do all of this, we used stool samples. We got the microbiome, we got the metabolome. It was 245, if I remember correctly, patients, and 300 patients from PBC. And so, first of all, we sequenced the DNA of the microbes, and then we started to compare them with control patients that don't have these diseases. So the key findings from this is that, first of all, we see marked dysbiosis in these diseases. When you compare PSC and controls, you can see that the diversity is lower. Same holds for PBC. And then we also looked at the individual species within the stool microbiome. So we found several species that are increased. For example, one very prominent is Escherichia coli. So this is a very known gut resident. It's no surprise. We found this bacterium in the gut, but it's increased in PSC. Another very important microbe is Klebsiella pneumoniae. Other studies have also correlated with this microbe with the disease, and but we also found species that were decreased. What's interesting is that when a species is reduced in one disease, it's also reduced in the other. We found that the gut microbiome of PSC and PBC has an increased capacity of having virulence factors. So these are proteins that do a lot of things. They are implicated in adherence of microbes in the gut. They are implicated in immune modulation. It's a very broad repertoire. But what is also important is that we found that a lot of the pathways in their genomes have either increased capacity or reduced capacity, which means that, for example, the microbiome of PSC can produce less vitamin B6. All of this could be used potentially as pieces of a puzzle to understand why someone is having PSC, and most importantly, how we can intervene, because we're trying to identify. So the goal of our overall study was to see if we can identify several bits and pieces that can actually be used for interventions, or to at least try and stratify the patients, because everyone has a different outcome. Having the metagenome and the sequenced microbes is a static snapshot, but this doesn't tell you anything about the actual metabolites that are inside the gut. So, for example, we found a reduced capacity of producing vitamin B6, but we also wanted to see if there is also reduced vitamin B6 in the gut. So we did this through the metabolome, and indeed we found several important molecules that were decreased, vitamins, and we also found some other molecules that were increased that are potentially implicated with pathogenesis or with liver damage. The last important finding is that when we tried to address if the microbiome actually correlates with advanced liver cirrhosis, advanced liver disease, we found several correlations. And most importantly, we found that the oral microbiome is actually increased in the gut. We found several species that are primarily of oral origin. They are increased in the gut of these patients, and then they are actually correlated with increased risk of hepatic decompensation, which means that the liver is damaged but can no longer compensate its function, and actually it's kind of a one-way street to liver transplantation. And of course, liver transplantation is not available easily. It's a very hard process. From these oral bacteria, there are some virulence factors, as I said earlier, that also are implicated in the hazard of having hepatic compensation. So, and especially the genus of Streptococcus, which is a very well-known bacterium from the oral cavity. And lastly, we also found that certain bacteria were protective against hepatic decompensation. So, and when we looked at what all of these bacteria have in common through the literature, we could see that they are butyrate producers. So, butyrate is one molecule that's known to be important for gut health and overall health. And we were very surprised to see that it's actually probably implicated in hepatic compensation. And then when we looked at the pathways again, we were prone to look at the pathways again, we saw that each of these subgroups of patients actually had reduced capacity of producing butyrate and through the pathways and also through the metabolites. So we speculate and we propose that probably microbiome level interventions of the gut will help PSC and PBC patients in the long run to manage these diseases.